Humanity has sent around 30 spacecraft and landers to the red planet since the space age began. Now we know which microbes might have survived the trip, says geneticist Christopher Mason, trundling across the surface of Mars. As you read, this is a remarkable machine, Perseverance, the car-sized rover that safely touched down on the Martian surface on February 18th this year, might only have a top speed of less than 0.1 miles per hour, but it carries a wide range of tools, instruments, and experiments that have already made some groundbreaking achievements. Included on board the 10 feet long rover is a machine that has turned a thin, carbon dioxide rich Martian air into oxygen, and a helicopter the size of a tissue box that made the first ever power controlled flight on another planet. The helicopter, called Ingenuity, has now made 3 successful flights, each longer and higher than the last. But did anything else come along for a ride with all this hardware? Could a trace bacterium or spore from Earth have accidentally been carried into space and survived the trip to make its new home on Mars? NASA and its engineers in the Jet Propulsion Laboratory have precise and thorough protocols to minimize the number of organisms that might inadvertently hitchhike on a space mission. Internationally agreed standards guide how rigorous these protocols should be, and NASA meets and in some cases exceeds them. Yet 2 recent studies highlight how some organisms might survive the cleaning process and also the trip to Mars, and also how fast microbial species can evolve while in space. First, let's start with the process that was required to build the Perseverance rover, as well as most spacecraft made in JPL's spacecraft assembly facility. Their spacecraft are painstakingly built one layer at a time, like an onion, with everything cleaned before it is added. These methods limit the bacteria, viruses, fungi, or spores on equipment to be sent on a mission. Spacecraft are built rooms with air filters and strict biological control procedures. These are designed to ensure that only a few hundred particles can contaminate each square foot, and ideally, no more than a few dozen spores per square meter. But it's also almost impossible to get to zero biomass on a spacecraft. Microbes have been on Earth for billions of years, and they are everywhere. They are inside us, on our bodies, and all around us. Some can sneak through even the cleanest of clean rooms. In the past, tests for biological contamination have relied upon the ability to grow, often called culturing, life from samples swathed from equipment. The newer methods used by my colleagues and I take a given sample, extract all the DNA, and then shotgun sequence it, as the term implies. It is like taking a shotgun to the cells of a sample, blasting them into billions of small DNA fragments, and then sequencing each piece. Each piece or sequence read can then be mapped back to the known genomes of species that are already present in the sequence databases. Since we know we can sequence all the DNA that is present in the clean rooms, and not just the ones that can be cultured, we can get a more comprehensive look at what kind of microbes can be found in the clean room, and if they can even survive the vacuum of space. In JPL's clean rooms, we found evidence of microbes that have the potential to be problematic during space missions. These organisms have increased numbers of genes for DNA repair, giving them greater resistance against radiation. They can form biofilms on surfaces and equipment, can survive desiccation, which is forced extraction of all moisture in the body, mummification, and things of that nature, and thrive in cold environments. It turns out that clean rooms might serve as an evolutionary selection process for the hardiest bugs that then may have a greater chance of surviving and journeying to Mars. These findings have implications for a form of planetary protection called forward contamination. This is where we might bring something accidentally or on purpose to another planet. It is important to ensure the safety and preservation of any life that may exist elsewhere in the universe, since new organisms can wreak havoc when they arrive at a new ecosystem. Humans have a poor track record of this on our own planet. Smallpox, for example, was spread on blankets given to indigenous people in North America in the 19th century. Even in 2020, we have been unable to contain the rapid spread of the virus that causes COVID-19. Forward contamination is undesirable from a scientific perspective too. Scientists need to be sure that any discovery of life on another planet is genuinely native there, rather than the false identification of alien-looking Earth-grown contamination. Microbes can potentially hitchhike their way to Mars, even after pre-launch cleaning and exposure to the radiation in space. Their genomes may change so much that they look truly otherworldly. We have recently seen that novel microbes have evolved on the International Space Station. Although NASA's engineers work hard to avoid introducing such species to the Martian soil or air, any sign of life on Mars will have to be carefully examined to ensure it did not originate here on Earth. Not doing so could potentially spark misguided research into the universal features of life or Martian life. Microbes carried into space can also be a more immediate concern to astronauts, posing a risk to the health and perhaps even cause life support equipment to malfunction if they become gummed up with colonies of microorganisms. But planetary protection is bidirectional. The other component of planetary protection is avoiding backwards contamination, where something brought back to Earth presents a potential risk to life on our own planet, including to humans. This is the theme of many science fiction movies, where some fictional microbes threaten all life on Earth. But when a NASA and European Space Agency (ESA) mission is launched towards Mars in 2028, it could become a very real consideration. If all goes according to current plans, the Mars Sample Return mission will bring back the first Martian samples to Earth in 2032. Past studies have indicated that Mars samples are very unlikely to contain active hazardous biology, and Perseverance is looking for any signs that might have been left by ancient microbial life on the planet. But NASA and ESA say that they are taking additional precautions to ensure all samples returned from Mars will be safely contained in a multi-layered isolation system. There's a chance, however, that if we do detect signs of life on Mars, it could have come from Earth in the first place. Ever since the first two Soviet probes landed on the Martian surface in 1971, followed by the US Viking 1 lander in 1976, there likely have been some fragments of microbial and maybe human DNA on the red planet. Given the global dust storms and trace amounts of DNA that might have gone with these spacecraft, we have to be sure we don't fool ourselves that the life we find isn't originally from Earth. But even if Perseverance or the missions that preceded it did accidentally carry organisms or DNA from Earth to Mars, we have ways of telling it apart from any life that is truly Martian in origin. Hidden within the DNA sequence will be information about its provenance. An ongoing project called metagenomics of subways and urban biomes, is sequencing the DNA found across more than 100 of the world's cities. Researchers from our lab's Metasub teams and a group in Switzerland have just published these and other global metagenomic data to create a planetary genetic index of all sequenced DNA that has ever been observed. By comparing any DNA found on Mars to sequences seen in the clean rooms of JPL, the subways of the world, clinical samples, wastewater, of the surface of the Perseverance rover before it left Earth, it should be possible to see if they are truly novel. Even if our exploration of the solar system has inadvertently carried microbes to other planets, it is likely they will not be the same as it was when it left the Earth. The trials of space travel and unusual environments they encounter will have their mark and cause them to evolve. If an organism from Earth has adapted to space on Mars, the genetic tools we have at our disposal could help us figure out how and why the microbes change. Indeed, the strange new species recently discovered on the ISS by scientists at GPL in our lab included some of the similar adaptations as those found in clean rooms, including resistance to high levels of radiation. As more and more extreme biology is cataloged in a program called Extreme Microbiome Project, there is also the potential to use the tools in the evolutionary toolbox for future work here on Earth. We can use adaptations to look for new sunscreens, for example, or new DNA repair enzymes that can protect against harmful mutations which leads to cancers and aid the development of novel drugs. Eventually, humans will set foot on Mars, carrying the cocktail of microbes that live on our bodies and inside our bodies with them. These microbes too will likely adapt, mutate, and change, and we can learn from them too. They may even make life on Mars more tolerable for those who go there, since the unique genomes adapting to the Martian environment can be sequenced, transmitting back to Earth for further characterization and then utilized for therapeutics and research on both planets. Given all the planned Martian missions, we're at the shortest edge of a new era of interplanetary biology, where we will learn about adaptations of an organism on one planet and apply them to another. The lessons of evolution and genetic adaptations are inscribed in the DNA of every organism, and the Martian environment will be no different. Mars will write its new selection pressures on organisms that we will see when we sequence them, opening an entirely new catalog of evolutionary literature. This is not just for idle curiosity, but rather a duty for our species to protect and preserve all the species. Only humans understand extinction, and thus only humans can prevent it, which applies today as well as it does in the billions of years. When the Earth's oceans begin to boil and the planet becomes too hot for life, it is inevitable that some transfer of human and microbial biology will occur when we start to head off towards the other stars. But in that case, we have no choice. Eventually, careful and responsible forward contamination is the only way to preserve life, and is a leap that we must begin to make over the next 500 years. And this has been possible star travel on Maybe I'm Rambling. Don't forget to like, share, subscribe. Tell me what you think in the comment section. Peace out.